In the news, if you heard yesterday, I think it came that Margaret Thatcher died of stroke. So this another opportunity to speak about stroke and what we can do, not to cure it, because I think it is not possible to cure stroke at this time, but at least to diminish or alleviate the effects, post-stroke effects, if we are lucky enough to survive it. And I keep trying for maybe now 15 years, saying we should use for this purpose, for therapies, the right model, that is aging animals, because aging is the key or the most important factor for stroke. And I do not know how high the incidence of stroke in Spain is, but I can tell you in Europe the rank is like this. First is Russia. They have the highest incidence of stroke. And the second place is the former Yugoslavia, now Serbia. And third ranking is unfortunately Romania. So all Orthodox countries, or Slavic countries. I don't know if it is any connection with religion or with vodka, I don't know. So this is just a short overview of incidence. This is how it looks in humans. And maybe some of you know there is a stroke committee and meeting every year in the US. And every time they issue some key points about therapies of stroke. And there are many criteria, but one of the major criteria is to test any therapy or at least strategy on aged animals. I will show you shortly why, and we had also a discussion with colleagues today. We know all that if you have a stroke in young animals, they do recover after 1 week, as would anything happen. But this is not the case in humans. You know, the most debilitating conditions are after stroke. This is not just a matter of money, but for the families, a big problem for caring for these debilitating people. And I would like to illustrate you what does it mean to have stroke in young animals and in old one. This is young animals, no stroke. The job is to maintain balance of this wooden beam. The stroke is from this side. So this is usually we do it on the left side, or the right side. It just happened to start the experiment this way, and we kept it on. There's no reason to do left or right. And this is an old animal with stroke. So the lesion is again here. So this is a big difference. It does not matter if we find genes or proteins or aspirin working very nice in young animals. They do not work in aged animals. So we set up several years ago and asked the big difference. It's just not just one gene or one protein. There are so many changes after stroke. It's pointless to look after some miracle, some drug which is used for recovery. So we ask actually how many genes or what groups of genes will be changed after such a big injury to the brain. And we did it in a collaboration with a group from Würzburg, genomics group, and we sent simply from our experiments RNA, and they did ask for the genomic analysis. And it looks, the general genomics, what does it tell to us? On the left, we have this correspondence analysis. That means what are the major factors which contribute to the variance of stroke atomic. And this is, these numbers are here. That means the biggest contributor, this is over 50%, to the variance is the lesion itself, the stroke. The second most important factor is about 25% of variance is due to time after stroke. And only after these two, lesion and post-hoc time, we have the age. So a little bit disappointing. Age is not the most important factor for gene expression after stroke in aged animals. If you look at sources of variability, so we pick up these two, stroke and post-hoc time, and have this diagram here in versus naive. And we have this green is denotes genes change in young, and red are genes change in old. And this cluster of genes corresponds to the naive animals. So if we look at these two, we see really there are differences between young and age, but not as much as we have expected from physiology at least. So this number, 12%, is not so high, but in physiological terms, actually, we saw a difference. Actually, we have expected more, actually. So physiology is not just the number of genes. Should be more than numbers. Should be some qualitative difference. So we look a step further at the Venn diagram and showing the number of common upregulated genes. With green, we have the gene that was just in young change, and in the blue one we have the number of genes change in age rats. And we see by simply expecting this number at day 3 and 14 after stroke that the number of common genes did not change too much. But what is different is that from day 3 to day 14, the number of upregulated genes increased from 150 to practically twice as much, but in young rats it decreased. That means we have a divergent gene expression. We have much more genes that increased after stroke from day 3 to 14, but at the same time this gene decreased in young animals. So that means once again changes in physiology do not reflect necessarily the numbers, but these numbers could be increased or decreased, so it's much more complex, the genomics analysis. If we look at the downregulated genes, again we have just few genes which are common, 13 or 21, and here in terms of downregulated, it happens just the opposite. If we have an increase here in number of upregulated genes, look here in aged animals, they decreased. So just again opposite pattern of expression. Then these comments or numbers are illustrated in this diagram. Look, if you look like this, so we have opposite changes in the pattern of gene regulation. And we did an innovation in this paper. We trying to represent patterns of gene expression after stroke, assuming that there is no change in non-lesion animal. And if you look at these two diagrams, what strikes us is not the number of permanently upregulated genes, but the big changes are in the number of these. That means temporarily upregulated genes, the number of delayed upregulated. You remember in the previous diagram, aged rats had a lot of upregulated genes late after stroke, 2 weeks. This reminds us of aging. They respond with delay at everything. But then they respond. So the magnitude of response in aged animals is not lower as you could have expected. No, it's not lower. The aged rats respond very robustly to the lesion. But the problem is the regulation or the unfolding of gene expression in time is not regulated as in young rats. It's not a question of number, but how these numbers are distributed over time. And we did it for several categories. We looked at physiology in young and neurogenesis, synaptic plasticity, and metabolism, embryonic development. And maybe one interesting thing is if you look at genes which are required for embryonic development and brain plasticity, you see what is missing here is this category. There is no downregulated genes, just either upregulated or delayed or temporarily, but no downregulated genes. So to summarize this part, this tells us, and we analyze by this technology, using this technology, we found about 2,000 different transcripts. So this means that any single therapy is going to fail. We cannot use one therapy to treat or cure stroke because of many changes in all categories. Metabolism, physiology, plasticity, all are changed. So we have to, if we want to have success, we have to change our philosophy to treat stroke. Except we want to invest more in prevention, which would be, of course, better. So gene expression for proteins which are required for synaptic plasticity is actually down-regulated. Does not matter if it is young or age. That means the message is the young rats are not better in upregulating genes which are required for plasticity. So they do not better because of plasticity. They recover, as I said, maybe 1 week, but for other reasons. The reason is not increased plasticity in young rats. Not even young rats can replace the dead tissue. The same holds true for dendrites. So several years ago, a colleague of mine, Zal Kokaya from Lund, published a review saying actually, or summarizing the fact that the age brain actually do respond to stroke and do produce these stem cells. But his model is not a cortical model. His model is striatal. That means for these stem cells to reach from this subventricular zone, the damaged zone, which is a few micrometers apart, is no problem. The problem is if you have stroke here, the cortex, which is more normal or mostly the case, these stem cells or progenitor cells are indeed produced in huge numbers. Does not matter if it is young or old. Each rat produces the same number too. But it is not possible to overcome this barrier. So for this model, it's nice to say you have neurogenesis, and neurons will migrate to the injured area and do some repair. Could be true for stroke in striatum, but not in cortex. So for this reason, we said, okay, we have neurogenesis, maybe not so strong as in young, but we want to stimulate it for therapeutic purposes. And our model is a cortical one, so we make a cranioectomy, expose the MCA, which is mostly affected in humans, and using a small hook like this, we pull it, and having a laser Doppler, we say, okay, now it's closed. And we keep it for about 90 minutes, and then we release it. That is reversible stroke. With this model, you can operate aged animals. With the filament model, we had a discussion today, the mortality rate is very high, but not in this model. Here they survive up to 80%. So it's actually very good suited for this for stroke. And what is the most important thing? We do a lot of behavior. As I told you, and I keep saying, you can find many genes and proteins changing, but the ultimate effect is the physiology. If you do not have an improvement after treatment, it's no use to you to do it any further. And we started with this molecule for a simple reason. It used to be used in the clinic in clinical trials phase 2, and with some encouraging results. And I met the group leader from the clinical study and said, okay, but we did not look in the in the age rats. So we did this more study. And we treated the half of the human dose. In humans, you use about 30 microgram per kilogram. We use it 15. Not because we didn't have, but we said to start with the lower doses. And we had some positive effects. This is important, as long as the treatment was done. That means about 2 weeks. In rotating pole, we have some recovery of function in the maze, inclined plate, and maybe the most important finding of this, the mortality rate was decreased by this drug. And it was amazing, none of the aged animals operated or died in this experiment. So that was the first conclusion, that granulocyte colony stimulating factor reduces the mortality rate after stroke. And we made a connection, if you want to publish in good journals, sometimes we have to find, besides the main effect, some molecules. So we look at neurogenesis, and it is indeed increased, but not in the cortical area, rather in the dental gyrus of the hippocampus, which is actually an area remote from the main lesion. But you had more neurogenesis. There is no direct connection to functional recovery, but it was an additional data to have a better publication. Then, after we did this study, we know this factor, granulocyte cell colony stimulating factor, is a chemoattractant. It simply attracts many things to a lesion. So we said, okay, we should try the combination, and had a consortium, or the funding from Germany, and my group was the leading one because all sent us the stuff, stem cells or GGSF, and we did the experiment again in age rat. This time was a big experiment. That means 20 rats per group and 4 groups. And the design was like this. We trained the animal. We always train in advance, about 2 weeks needs, do we need to train the animal before stroke, because if you do not train, they do not know what is the task. Then we have the occlusion here, and we started the therapies at 6 hours post-stroke, did the first MRI at 3 days post-stroke, and the second MRI at about 45 days, and has a tissue analysis at 60 days. All this time we analyze weekly for the animals. So this is, we have the spectrum only. We work only with male rats. And the age, we had also a short discussion today. We work with 20 months old rats, that is in humans about 60 years. So not very old, because there's no point to find something or therapy for somebody who is already 80, works anyway any longer. So we keep saying if something should be done, then at an age which is reasonable. So 60 years old, especially for us getting older, is better. And the control group had just glucose. So we made the first combination. This was alone or with bone marrow mononuclear cells combination, or with bone marrow from mesenchymal cells from rat. We had a small group with cells from humans, but just for labeling, not for therapeutic purposes. We gave them via the jugular vein. Currently we administer intrathecally, which seems to be more efficient. We have better pictures, but this one is just to show that the human cells do reach. This is the infarct area. You see it very easily. They enter either via ventricles or because of the damaged blood vessels. What happens with survival? There is no significant difference this time, but the number of deaths was anyway very low, which is almost is normal. You don't need any treatment to have to dead rats per 20. Here is a summary. We have the MRI at day 3. We have here a big edema, which and at 45 days is just remaining stroke. That's so to speak. So this is another objective of the project was to compare histology with MRI. You see, it's pretty similar with histology, and this is Nguyen staining. And the bad or the disappointing result, we had really no reduction in stroke volume. Does not matter if treatment or not, no significant at least in any group. So stroke lesion was not reduced. Then we look at several functions. This is the water maze. And if you look at a path before stroke, and then they are trained, they reach rapidly, or more rapidly the platform, and after stroke it gets bad, but then it gets better. We had some improvement in the water maze test, but that was short-lived, about 21 days. This reminds me of the first treatment with GHSF. The good effect was as long as we kept the drug, the animals on drug. If we take the drug out, the effect is vanished. Then we look at sensory test, and here, to our surprise, the best group was granulocyte colony stimulating factor alone. So the combination treatment again did not improve, contrary to our hypothesis, did not improve the function. I would like to say there are hundreds of or thousands of papers always maximizing some minor effects. This is not good for stroke research. If you don't find a good positive effect, which is obvious, don't try to sell your data as a very good one, because this is one of the reasons over 1,000 drugs have been positively tested in animals, but later none of these 1,000 worked in humans, because many people tend to improve or to sell better the results. We look at this rotating pole. For me, it's one of the good tests. And again, the group, so not a combination, but this old factor was had showed the performance over all testing period. Other had, but again, just sporadic for a short time. So the consistent result actually, or improvement, was again shown by this peptide. What about neurogenesis? I told you all rats really do produce a lot of progenitors. And we stain it here, double coating, which is shown in red, and with green it is actually the endothelial cells labeled by BRDU. The problem is none of these progenitors is labeled with BRDU. That means there is no direct connection, or better to say, this BRDU, this double cortin stains better about 2 weeks after lesion, but this is continuous. That means at the time we injected BRDU, that was during the first 2 weeks, we had not too many BRDU, or if we had, we lost them. So this is 2 months after stroke. So at 2 months, yes, we have neurogenesis in the subventricular zone, but it's not BRDU label, so we are not sure if it contributes to anything, but it is there. So look at the impact. And we had something which is looking like, let's say, stem cells, or have a similar morphology at least, but this was true only in the group where we have the combination with mesenchymal cell and this peptide, not in other groups. And we had here, which are also PROX positive. So we did here quantification, and really in group C, that means combination of mesenchymal and granulocyte colony stimulating factor, we had more of these cells. Otherwise, most of the BRDU, maybe some of you know, actually goes into the blood vessel. So we went a step further and had and asked and this is a key question many people ask it: what is better, to have more neurons before lesion or to produce them after a lesion? For this, and we did some experimentation to answer this question, and stimulated the rats long before the lesion, about 2 months. And we did it either by stimulating the ear. We have a very good neurogenesis if you give a short electric stimuli, or by injecting a chemical which is used for epilepsy models, pentylene tetrazole. And this is a flow diagram. So as I told you, we injected or treated long before stroke and labeled the cell with BRDU. So we are sure at this time point we have a lot more neurons in the brain than in the normal brain. Or some groups were stimulated after 1 week after stroke. So we produce in this experiment more neurons after stroke. And we have again our testing. So the sensory did not change. It does not matter if we have more neurons before or after death, let's so speak. But in the inclined plane, we did observe a good effect. This inclined plane is like this. You have the rats on a plane, and you keep rotating at an angle, and at a certain angle they fall down. So this is a measure of ability to maintain position of this inclined plane. And what was the first indication that more neurons after stroke is beneficial. So it's no use to be a smart guy before stroke, in other terms. It's better to have more new cells after stroke. And this is true also for memory test in the radial maze. So rats being treated or with increased neurogenesis after stroke have better recovery. But overall, I should say age rats never recover to the value before stroke. They recover maximum to 60-70%. Young rats recover 110% for other reasons. We did some quantitation. Stroke volume is not so important. There was a study, in function terms. There was one or more studies years ago showing that if the stroke is large enough, that means more than 5%, which is not very much. Our rats have about 30%. So if you have stroke more than 5%, you do not see the correlation between function and lesion stroke. So if you want to see the correlation, please very, very small stroke. If you have big stroke, forget the correlation between function and volume of the stroke. So we did many quantitation, which is for paper purposes. And I want to stress once again, this is a lateral ventricle. So you see a lot of double quartile cells emanating from the wall of the lateral. So you have really also in old or older rats, middle-aged rats, a lot of neurogenesis. But still we do not know where they end. They are there, they migrate, but in my opinion, they do not overcome the corpus callosum barrier. And even more, with green we label the axons. We do have some chaotic axonal growth, but this is in every direction. Normally, if you look at the normal brain, you see the axons parallel, so you have an orderly structure, but not after stroke. You do have many, but they are not organized. Then another issue is the scar, which is inhibiting, according to many people or groups, the axonal growth. And if you compare young versus old, this is again a major difference. Shortly after stroke, you have here a strong scar in aged animals, but in young animals it's still formatting. It's not there. There later after 2 weeks, no big difference, but the difference, at least in histology, you have in the beginning. Very rapid formation of the glass scar in aged rats. And what is the major contributor to the scar? Actually are the disintegrated blood vessels. Most of the cells contributing to the scar are detaching practically. This is a blood vessel from fragmented blood vessel. And I have here another picture, how it starts in the cortex. This is the corpus callosum. If you look at many tissue from stroke, you see in the beginning they are like corridors, starting from the corpus callosum to the cortex, and these are inflammatory cells. So with increasing time, we see that they free nestin cells, all detaching, like here, from the wall of the blood vessels. And this is maybe more clear. This is a BRDO nuclei. This is nestin cells. This is with blue we have laminin, so this is a blood. So look how many cells are detaching from blood cells, and most of them reach this position. This is the scar. So the scar is consisting actually, if you look at these two, from cells detaching from the wall of the blood vessel. At least some of them. So we made finally this cartoon. This is a lesion in the cortex. This is our capillaries. This is blood vessels. Some cells are leaking through the damaged blood-brain barrier, but many are detaching from the damaged or fragmented blood vessel and reach this or contribute to the glial scar. This is inflammation. The last point of my presentation. This is no discussion how damaging could be, first of all, because of edema in humans. And you can see on this picture is again at 3 days what means inflammation in old rats. So how much phagocytic cells we have compared to young one. So this is again a major difference, how rapidly the scar is built up and how intense is the inflammatory reaction. So then we reason, okay, we have a lot of inflammation, we should try to reduce it using hypothermia. We were one of the first group trying hypothermia, but not physical hypothermia, but gaseous hypothermia. That means, this is again the scar. I just want to show you at 2 months after stroke, this scar region is not dead or did not stabilize. This is, look, this is a scar, the red cells, but here we have a lot of inflammation going on, even after 2 months of stroke. So we try to reduce inflammation using hypothermia, and we did this experiment again in young rats, and we were inspired by a science paper showing that gas, hydrogen sulfide, induces in mice a state of like hibernation in animals. So they, if you give 80 parts per million, so very few molecules of this gas, animals do not drink, do not eat, they look like freezing in time. And they reach about 31, 32 degrees. And we kept the animal for 2 days after stroke under these conditions. And here is the amount of expired carbon dioxide, which is normal, goes down. The more of this stuff they have. And we measure cortical activity using telemetry, and here we have the temperature. So as I told you, we reach about 30 degrees, but not at once. It takes about 8 hours to reach, but then it is a rebound. So you cannot keep them 1 week in this. The temperature, even you have did not remove, we have some reduce the amount of this gas, but only to the half. But no matter if you reduce it or not, the physiology is recovering, so the temperature goes back. And the cortical activity, you see, there is a very good correlation between activity under normal conditions, let's say, and in hypothermia you have a very low activity. Again, we measure the infarct this time shortly after hypothermia at day 2. And indeed, with this procedure, it is possible to reduce infarct volume. And I tell you, the results are really impressive. Not by only by MRI, but also by histology. The major difference, if you look at the histology, is actually maybe not the area itself, but the number of dead cells. Not so many under hypothermic conditions. And this is a quantification, again the comparison between MRI and histology for infarct volume. Here we have several types of showing the electric activity as a gamma or normal after stroke. And by this occasion, we did the proteomic analysis and looked to identify genes and proteins which are important for hypothermia, and first by arrays, and found this annexin 1, and then by proteomics, which was actually a confirmation of what we have found. And this is confirmation of genomic studies by Western blot and two-dimensional electrophoresis. This is a quantification. So we have a large reduction in the hypothermic group, in this annexin 1. What is good for? So we phenotype it. This is a normal staining, chemical staining, and here is by fluorescence. We tried to phenotype this molecule, and we found that it co-localizes with polymorphonuclear cells. And exposure to hypothermia actually reduces the number of polymorphonuclear cells. It is one aspect of hypothermia. We know there are hundreds of them, but this is what we have done until now. And our current grant application tries to combine more, and I finalized with this. We tried to apply hypothermia in the first 1 day, and then try to kill the phagocytic cells by using hyperthermia, using laser technology, and then after 7 days try to restore some tissue by applying mesenchymal cells which are attracted to the lesion site using magnetic field. This is so-called Oro-nanomed approach which we are currently trying to implement. So I have several conclusions. The only treatment which really helps, but in the beginning, I do not want to go into more details, is hypothermia. It really reduces inflammation and lymphatic volume. Still here, there are many things to be optimized. And one central question is, what is the difference between physical hypothermia? You know, in clinics you use ice to cool the bodies. Patients do shiver. There are many other things. So we have to define the therapeutic window. So there are a lot of things to be done, but it certainly is work. We think the aged brain is still capable of repairing, but needs to be stimulated, had to be stimulated. One problem is this fibrotic scar, but I do not see at the moment any pharmacological means to get rid of it. And what is the most important, we think that the multimodal approach has a better chance to not to cure stroke, but to have less damage after stroke. And I want to thank you for patience. to listening to me.